So I want to welcome everyone to this discussion of estimation and estimation methods and approaches within OHDSI. I think I know everyone on the call, but just in case, my name is Marc Suchard. I'm faculty at UCLA in biostatistics. I've been engaged with the OHDSI community since its beginning, and I'm very interested in what we can do as a community to improve causal estimation from observational data. As Craig mentioned, this is not an opportunity for me to present. I'm basically just here with a couple of helpers, including Fan Bu, who's on, and Kristen Koska to help me mediate a discussion. I thought I'd kick off by just putting a couple of discussion points in the chat to help get us going. And we can use those as kind of starting off points for a further undirected, more useful discussion. So maybe one place we can start is a little bit of focus on what has OHDSI's successes been in terms of our estimation approach. We very rarely talk about our successes, but that gives us a good, foothold to then move on to where are we really failing, and as a community can do much better. So I'm going to try to shut up now and help get a discussion moving.
Hey, Marc, I have an easy one. Show of hands, how many of you have used Cohort Method, R library? In any form. Someone built it for you, you wrote it yourself. Good, I see lots. Okay. So good. How many of you built your own?
Like run it, you mean, not build it.
No, built it from scratch. Like did not inherit it from Marc Suchard.
Definitely Christoph.
Yeah, Christoph might be a
for reasons that we can certainly get to when it comes to weaknesses and limitations.
Just curious, that helps.
Yeah, so cohort method is one approach or one design for estimation, but I think the community has done quite a bit of work in general, perhaps not as causal related, but estimation types of questions, starting from starting from kind of estimation of incidence of different cohorts, including kind of drug utilization, and the starburst type of work. But we certainly haven't focused as much as we could in estimating population level estimates that are more in the descriptive category than causal category.
I mean, I think if you take Patrick Ryan's framework for reproducible evidence, and you go through each of the pieces, being able to reproduce your experiment, someone else doing it, similar population, different population, et cetera, and triangulation. It's about different databases, different researchers. So I think what OHDSI is uniquely doing is bringing in a community and addressing causality through a community and pulling in those resources. And I think if you take the legend 10 principles, we've reorganized them into two, great commandments, which are, instead of the 10 commandments, which are, verification and openness. So I think OHDSI's done really well in openness. So pre-publication, pre-specifying your protocol, sharing your source code, looking at your diagnostics before you look at the results, sharing the results before you look at the results so that you can't change things when they don't match your expectations and all those things, and putting everything on the internet so you don't have publication bias. On verification, I think we've done really well on diagnostics. Everything is about how are we going to show that this is, and it's not perfect, sure, manual chart review of 1 million cases would be better than Phi-Valuator, but we can't do manual chart review of 1 million cases, so we have Phi-Valuator and all the other balance and negative control diagnostics. So I think we've been in equipoise. So I think we've been really good at coming up, and the whole mindset is doing this large scale so that you have, you know, so you can generate those diagnostics basically. So that's kind of how I view the number one part. And being honest, and like when things don't work, we actually admit it as opposed to proving that it works anyway. And it's striking, actually, just 2 recent grant reviews. Not recent, but well, one recent and one older. One is like, if you do this in multiple databases, those database results might conflict with each other. You know, yeah, so we shouldn't do it in multiple databases. And remember, Pecori awarded the award to someone who's doing the entire analysis on a single database, and they won't have that problem. And in fact, if you do no study, you won't get bad results because you'll have no results. You know, I mean, so like, and the other one I was thinking of is never share your source code because it'll reduce your reliability because it confuses people. You need to hide your source code and just say what it does instead of actually disclosing your source code. Like that, it's not even not beneficial. It's actually, you know, harmful to share your source code. And this is what we're fighting. So I would say on number 2, our major limitation is we don't seem to be able to get the word out, and we seem to be fighting against this crazy battle.
So are we having troubles getting the word out because the ideas are so diametrically opposed? And, you know, that might argue, you know, perhaps we need to be the individuals with the, you know, with some sort of olive branch or you know, a huge statement of inclusivity.
And I think the metrics of science and publication, despite the, you know, team-based, collaboration-based, and so forth, are still pretty locked into, you know, what an individual can produce in many ways. And somehow the, you know, while we produce these amazing studies that are high quality, like it's a step up to do an individual thing and then say, now I'm going to replicate it across a whole network and do all that. So somehow if we could make it easier for the individual to be able to do that, who's, I don't know, I'm just shooting from the hip here, but somehow if an individual can get that credit and that even as the network, you know, I don't know. There's so many people on these papers, and still first and last author is still the bookends that seem to matter in academia. It's like, how do you deal with that? And you know, I just published a paper with 42 authors, right? It was let's face it, there's a serious amount of social loafing that happens with that. And so I think there's some downsides to the really big stuff too. Anyhow, some vague thoughts. Take it where you like.
I think a lot of it is like what you asked, George. It's changing behaviors, right? We're changing behaviors of like, what does that mean when we want to talk about like being able to do like science across different institutions and like in the context of the evidence that we're generating and like the impact on for decision making. So we're just like in the beginning of like just like scratching the surface to try to understand, like bring the word out to the community of all the researchers that like there are best practices to do that. So thinking about the whole concept of reproducibility, it's nothing new, right? Like it's been talked about like in a lot of contexts or all these like sort of guidelines, but then it's still so unclear and it's barely touched when it comes to practice. Like very common, like, I mean, like there's all this effort that is going on, and ISPI just recently just put out like sort of like a white paper or a paper about recommendation on reproducibility, and still the recommendation doesn't tell you anything, but tell you what is your inclusion criteria and exclusion criteria. Can I reproduce it based on that? Like, no, of course not. But I think this is a responsibility for us to start like talking about like what does reproducibility mean in practice when we want to actually do it. It's hard. It's a completely new topic, and it takes a lot more work to just make the entire community like follow this. And in some sense, sort of like remind me of like a lot of the guidelines, like Consort, Prisma, like how like reporting and best practices will actually lead to doing the good work. It's just like sharing some thoughts and like a lot of the work that we have to do still to just like spread the word, so it's not just us.
You know, it's.
Go ahead, George.
Or I was just going to say, you know, I think a lot of those guidelines that you have to fill them in, and then like you get a pass. Like you did the world's stupidest study, but because you filled out Prism, it's like, oh yeah, that's good. They did, look, they filled it out. It's completely filled out. I've never seen it so filled out. Meanwhile, like it's a ridiculous assumptions into it. The other pet peeve is when someone who published a paper with like manually like selected confounders, and they kill me because how do you know there's no unmeasured confounding? Like, I don't know, how do you know there's no like you just know, the structural causal model perfectly, you know everything except the hypothesis. If you know so much about that hypothesis that you know the full structural causal model and there's nothing else there, then you probably know the answer whether the hypothesis is true or not. You don't need to do the study. Like there has to be some ignorance there. Why is that the only thing you don't know? There's such a complex thing here, and that's the only thing you don't know. You probably know that one too if you know everything else. I think though, but I think one thing we could do, you know, we tend to like publish a paper and move on and publish a paper and move on. But if you take on one of these hypotheses and follow it over time in the literature, maybe we need to do that more, because it is complicated and we do get things wrong. And we, like when we did our study in philosophical transactions, you know, we picked 5 hypotheses. One we knew we were going to get wrong, and we got it wrong, unfortunately. We predicted we get it wrong, and we got it wrong. So we get things wrong. That was a bad one, you know, bad channeling bias that did in fact come through. So, you know, I've been following since I was the first author on the hydroxychloroquine, hydrochlorothiazide chlorthalidone paper. And like there's more stuff going on, and it's interesting to follow. And people have like, you know, still are not convinced, despite ours not being the only observational study that says, you know, be careful with chlorthalidone. But it's got a living, like you have to keep at it. And so maybe one of our faults is we publish a paper and we move on, and we publish a paper, move on, and we go to the next thing. And now let's do let's solve diabetes. We solved hypertension, let's solve diabetes. Well, maybe we didn't solve hypertension yet. And Yale is working on the next iteration. So we're not completely moving away from things. But, you know, maybe some of these things just have to be wrestled to the ground a little further.
To, you know, flip our flippant statements about, you know, Prism checkboxes on its head, you know, an alternative direction that we could consider is drafting and marketing what the Prism equivalent would be for large-scale reproducible observational studies. You know, at the point we've, you know, there are some principles. Principles are hard to follow. You have no idea, can I actually check the box or not? You know, so maybe we do need to reduce it down to a set of checkboxes and market it in the right communities where people see, okay, actually I can go through and I can check these boxes, and now I do feel a little bit better about reproducibility. Like one of the checkboxes is I actively tried to reproduce this in, you know, in a data set which I believe is different than, you know, the original one in which I imagined this work. You know, I have included several diagnostics. I have tried to look for evidence of unmeasured confounding. You know, but then it comes, you know, but you know, then perhaps those ideas then get used, and then of course we have the gaming. You know, I forgot your comment, George. It was fantastic. Look how well I checked the boxes.
So that's a good idea. I like that. I like that. let's
I heard
make
a
it so
rumor
that the people who are
Ozzy is working on that. She's got a secret yardstick that her and Paul Nagy have been crafting by hand. They're whittling the wood though. They could use some more supplies. But
Yes, please. But I think some of this is even by showing that these checkboxes is not ensuring the current available checkboxes. They keep coming. I mean, it's just impressive. Like, just show that like it's not ensuring reproducibility. What does that mean? That like I have your inclusion exclusion criteria, or I have your period, or I have 2 ICD codes. It's not going to give me anything. I don't know what's happening here. The codes will tell me this concept set. So I think there is a good opportunity to still argue and show like the sort of like the other side of like how we can actually ensure it, and keep doing it until it is accepted.
I think using check boxes as a laundry list of items that can support the validity of a study is only useful if it's actually linked to a piece of evidence that shows that it's good. So rather than just, in an appendix having a set of checkboxes, I'm thinking more of like, changing the reporting framework of these studies where these interactive apps that we've been using actually link to the items on a checklist, whether that's a checklist that already exists or whether it's an OHDSI best practices list that we come up with on our own. But the idea that each checkbox is just a yay, nay dichotomous thing is stupid. I think it's that each item on the list has to be evaluated with some evidence, like a diagnostic from our analyses. So I kind of I think changing the reporting framework a little bit to align with these, understood expectations of what constitutes valid evidence might be useful. And then I guess, now that I'm talking, and then secondly, we've got this pretty well understood concept of, we call it scientific best practices, but it seems to really be OHDSI community best practices. And I'm wondering in the experience of people with much more time in the field than me, it's like, how do you move from like a group's best practices to a larger community best practices? Like, there are lots of ways to try to assess and account for unmeasured confounding. We've got an approach. There are other approaches too, and neither are really accepted except within the sort of communities that fully accepted except within the communities that develop them. So, like, how do you take that step? OHDSI's made has best practices that we believe in. How does that sort of go beyond the community? I don't have an answer. I'm posing it to the rest of you.
Christoph, I can tell you want to chime in both in type and in verbal. So I think you get the floor.
Well, I'm trying to. So how there's living literature reviews, and we saw that emerged a lot in COVID. And in OHDSI studies, they have a beginning, middle, and end, and yet there's plenty more databases that could have been run. And over time, there's plenty more people who are, different interventions and outcomes and so forth are going to happen. So I'm wondering if a framework where evidence becomes a continuous process of, sort of showing, well, here's our ongoing understanding of risks and benefits of different things, and then we're not stuck with what's just cemented in the literature at a point in time.
I think the living research idea is something that we as a community are ideally or positively positioned to work on, because we've worked so hard in formalizing the process to make parts of it even high throughput, not meaning lack of rigor, but systematized. And there are a couple of efforts along that, a couple of very strong efforts along that line. One of the key objectives this year for the methods work group is to provide some guidance on what this type of living research would look like. I can speak to my own experience within the Legend initiative, and whereas some of the focus has switched towards diabetes, we have a very active group which is continuing to dig into emerging questions in hypertension. But we haven't sat down as a community and thought about what is the what are best practices for living research, right? When do you have to decide that we need to reevaluate the evidence that we've looked at? When are we satisfied with the number of data partners and the information in the data that they've brought together to answer a question? Besides, this is just, I contacted all my friends, and 80% of them are on board, so let's move forward. Well, maybe there needs to be some diagnostic to think about, is that sufficient representation or not? And then an important reward structure, which both in the community and at the kind of larger scientific community, that's harder to build for, to reward people who want to, work on what might look like the lagging end. I mean, I think that's a major weakness of the OHDSI community in general. Is it's a community of misfits, but one characteristic that most of them share is they really like to be on the leading edge instead of the lagging edge. So do we need to change our personalities, or do we need to find a good reward structure to encourage that type of important lagging edge work?
And I was in the private sector, running a software company, and you'd find that whenever you try to go deep into a given scientific area, you'd get to a specialization that required a lot of work for limited returns, and it was the platform builders that would try to do things in this generalized way, but would fall flat on going down those tributaries, kind of the fractal landscape of the complexity of a given domain. So what you say about that, we want to be in the leading edge, it's like that in a way. We're building these grand, you have this nice abstract notion, oh yeah, we solved that problem. But if you were to look below the surface, you'd see there's so much detail and subject specific stuff that often thanklessly, with a whole lot of effort, would be taken to work it out versus saying, yeah, we've done the big study on X, right? And now this idea, well, we're going to study all diseases and all outcomes and all drugs and so forth, and sort of, claim victory on that, or this phenotype February, which if you dive deep into each of these, there's tons of unfinished drop stitches where the long tail of working those things out is shall we say not well incentivized perhaps. So I feel we win as a community when we can have these big, bold, exciting studies and so forth, but there's a certain this weakness of, what was the phrase you used, Mark? The
rewards, or the lagging edge?
The lagging edge, yeah. So someone capture that. I wanted to say it before you said it, and you said it so well, and I just wanted to reinforce it. It's really a big issue, I think, in OHDSI.
There's just too much innovation.
Yeah.
I feel like there's two different aspects of answering the question, what we have achieved or successful of, and where are the challenges. There are successes in terms of best practices and methods and improving the science. And there are successes in terms of answering a clinical question or giving evidence for a clinical, just like diabetes or hypertension. And obviously, we have successful in one area and more or less success in another area. But it is like why you guys were talking, I felt like those are two separate things, because like if you focus on method, your measure of success is one, how much you move the science forward in your own local community, and that's all the innovation that we work on. And there's the other aspect of it, how much you influence other communities, regulators to adapt these things that you came up with, whether a certain a general concept or like a particular specific approach, how much adaptation has been happening. And I don't know how much you guys feel about how much influence you did over the last many years of the journey. And where do you feel that you did actually find, like there are meaningful successes in adaptation from other communities to the best practices that have been innovated, found and promoted. And then the other side is definitely, as Christoph was talking about, like how much we actually found, like provide evidence for problems. And I agree with you. I think many times we have big studies that we feel like we did everything, but oh God, there's so much details in everything. Like phenotyping is just one example. We did not finish 29 phenotypes. We started a conversation of 29 phenotypes. There's way a lot to be found. But yeah, so like I feel like there's two different things about successes and challenges.
One interesting point in the direction that this discussion has gone is, you know, when I was envisioning those questions, I was on a much finer grain. I was envisioning them on a much finer grain level. You know, we've on the level of we've done pretty good in sort of infrastructure for, you know, new user cohort designs. We got a lot of great infrastructure for, you know, self-controlled. We have very little infrastructure for simple summary statistics like incidence rates of you know, which things that people actually want, which are good population level estimates. And, you know, biggest weaknesses is we seem to be in a silent battle with another community on what constitutes rigorous causal inference methods, and why are we so resistant to adopting what or at least trying out what others have been promoting for years. But I'm happy the discussion went in a different direction, because I got to think about different things.
That's because we let George start, so he took us to a very high level thinking.
You know, to put an individual on the spot. Sorry, Nicholas, you're gonna despise me for this, but it is nice to see you in this discussion. And you know, as I was wondering how have you felt about points raised in this discussion, and you know, what are we really missing in terms of, you know, important drug safety monitoring, particularly from a European perspective?
Yeah, so those are interesting questions. I feel like I've been a bit far away from OHDSI for some time now, so it's nice to come back and here, and that's why I've stayed sort of silent here and sort of try not to get too much involved. But I think if I look back, and this is both OMOP and now OHDSI, I think where a lot of contributions has been in sort of systematically also looking at evaluation of methods and I think transparency and making things available, and that seems to still be happening. I think the calibration approach with the negative calibration has been a huge thing. I think we're still struggling with bridging the gap, I must say. I mean, in terms of how we go from pharmacovigilance and the scope we have, where we look at every possible drug, be it, you know, herbal medicine, over-the-counter medicines, you know, name it, to then be able to assess that. I mean, that's what we really want to do. Is we want to sort of close the gap from working with the case reports and scientific literature, and then having too big a step to where we go to the observational data and use that to inform the assessments. We want to much more integrated, so that we can have access to observational data much earlier in the process. And we had a great experience with Patrick Ryan, I guess 2 or 3 years ago now. He joined one of our signal detection activities, and we could do that there. And we had at least one really good example of how access to observational data really informed the signal assessment in a good way, and it changed it and strengthened it. But to go from that one example to see how often can we do that, on what scale can that happen, how reliant are we on good phenotypes being available to be able to address a question, or can we go with sort of a more crude approach? If there isn't a strong phenotype, can we just sort of map over from MEDRA to SNOMED and get something together with like an 80% performance, and how well can that support the questions we may want to answer? So this will be part of the Eden project, where we're leading the pharmacovigilance use case. So we'll try to address this, and we're in the middle of setting up to do that, hopefully during this year. And so I hope to come back with some more questions on that later on. But I'm not sure we'll get to the estimation part. I mean, in some respects, even some descriptive analysis can bring a lot of value to the assessment. So whether we go the whole way and actually do sort of a self-controlled case series analysis or cohort, with propensity adjustment to try to know, is this a solid association when we look at observational data? I'm not sure we'll go that far with too many combinations, but even earlier stages can be useful. So sorry, I was rambling a bit now. I don't know if I've answered your question.
No,
but I think
it was some nice insight, and it actually raised what appears to me to be a North American versus European divide within the community, which I'm interested in getting some insight in. Maybe Jamie can, since he's trying to swim in both worlds right now, can help give some insight. Estimation and causal inference methods and focus seems to be a North American activity. Even within the statistical community, whereas at least within the OHDSI community, Eden is, or at least those running Eden, are very focused on prediction types of problems. And how can we overcome that divide, remove that divide? I mean, I think it's there just because of historical reasons. Peter was very interested, at least very early in his career, on particularly in prediction. But is there a cultural difference that we're missing? Because if there is, then we can work at that cultural difference.
I'm not sure. I mean, I think we came at it from an estimation. When we were engaged in OMOP, that was all about estimation. It was from a signal detection perspective. So we're not at this sort of confirmatory stage, but it was about raising hypotheses. But our interest was then really to see, can we pick up signals of possible adverse drug reactions directly in the observational data? And we're using essentially inference methods like self-controlled cohort designs and so forth to try to do that and see which methods could do that best. Ultimately, we didn't really have much success in finding real safety signals, and part of it was we focused on our end, and this was not OMOP, but in other European projects, on UK primary care databases, and they just didn't have the broad enough coverage that we would need. I mean, a lot of drugs that were new were not on the UK market. A lot of the adverse events that were relevant to pharmacovigilance were not captured in the primary care data that we had access to. This is where we're much more excited about the Eden possibilities and broaden that OHDSI ultimately, where we can have data from much more different kinds of settings and from different countries and so forth, and see, can this now become a serious contributor to pharmacovigilance in this broader sense. But I mean, I'm very interested in the causal inference side and the estimation side, and I think that's ultimately what's relevant to pharmacovigilance. Of course, prediction can be important as well to know who is at risk, but I think the more common questions are, does this drug actually cause this adverse event? And then it is an estimation question.
No, it certainly is. I want to be mindful for everyone's time, so I'm going to close on a comment that follows on quite closely with a point that Nicholas, that I just pulled out of what you were talking about. Is a big strength of the OHDSI community is the ability to aggregate information across heterogeneous data sources. I think Eden is going to be a wonderful example of that. It's an opportunity to take what one, from the U.S. perspective, might say small data sets and aggregate them and aggregate the information. I think estimation has a very important role in that type of aggregation. And so in spite of reviewers saying, why would you possibly replicate this in a multiple database, we should certainly be focusing on the strengths that we get from this type of data aggregation problems. Thank you all for joining us today. It was a great discussion. We'll continue on with our community calls next week with something, which I don't remember what it is.
The CDM workshops are beginning next week and the week
Oh,
after.
that's right. The beat is on, or the heat is on. The beat is on for Claire. All right, and I'll see many of you at other activities throughout the week. Bye, friends.
Thank you, Mark.
Bye.
Thank you, Mike.